Okay, no, that's perfect. So before starting this webinar, we would like to thank everybody for joining us and for the interest you have in OncoKDM interpretation platform. Today, you are coming from all around the world to discover the most advanced way to turn NGS data into precision medicine. So on behalf of OncoDNA, I would like to say thank you again. During this webinar, you will have the opportunity to learn about interpretation of NGS data, and we'll try to answer those two questions. The first, why using an interpretation platform, and how those interpretation platforms turn NGS data to treatment decision and precision medicine. Then you will listen to the testimonial of Jordi Canes from Solti. If we have questions during the webinar, you can use the questions panel at the right side of your screen. We'll answer it after the end of this webinar. Before giving the floor to our speakers, I want to ask 3 questions. Of course, you don't have to raise your hands up, but just click on the right answer in the poll panel you will find on the right side of your screen. I will open the poll right now. The first question is, are you already using an interpretation solution? Answer yes or no. If yes, you can choose if you are using a commercial one or in-house solution. I'll let you maybe 15 seconds to answer this first question. Great. Let's move forward to the second question. The second question is, how many tumor genes are you screening? In other words, what about the size of your panel? I will display the second question, then you can answer. Once again, I'll let you 15 seconds to take the time to answer to this second question. Great, thank you very much. And the last one will be, what can be for you the most critical issue you can have when interpreting NGS data? Once again, I will display the question in the poll panel, then you can answer to it. Okay, good. I will close now the poll. Thank you for your answer. We will now start this webinar by giving the floor to Nicolas, who is our OncoKDM project manager. Thank you.
Hi everyone, so I'm Nicolas, I'm project manager for OncoKDM. I'm going to present you our web-based interpretation platform. So first of all, the reason for this platform is that nowadays we have huge amounts of data allowing us to understand exactly what we are dealing with and therefore finding the best way to fight it. However, it is becoming more and more complex to integrate all the data available and to use it efficiently. This is why we think that there is a need for tools like interpretation platforms, allowing this process to be smoother and leaving the practitioners more time to focus on the real complexities. So first of all, let me give you a brief introduction of OncoDNA. So OncoDNA is a Belgian company that was founded 7 years ago by the founders of DNA Vision. Our goal is to provide the oncologists with the right tools to clearly identify the tumor and to find the best treatment for a specific patient case. We started working with different types of kits based on solid and liquid biopsies, depending on the cancer type, its stage, and of course, the availability of the samples. These ones are out of scope of this presentation, but if you want more information, please don't hesitate to contact us after the presentation. So the usual process is that the oncologist sends us the biological sample, we sequence it at the genetics and pathology lab next door. Then we get the data, run them through our bio-IT pipelines, we analyze them, feed our database, add annotations, etc., and provide the oncologist with a complete report that spans across all the data that we received. By working with these kits, we created a huge knowledge database that we wanted to make available for hospitals and laboratories that are using NGS in-house. Therefore, we decided to launch OncoKDM, which stands for Knowledge-Driven Medicine, and which is a platform allowing you to interpret your data using our knowledge. We use sequencing data and integrate also other types of data that your lab might generate, and we provide you with a comprehensive report that spans across NGS, but also IHC, microsatellite instability, H&E stain. We look at the molecular pathways, etc. Well, I'll show you more later. So why would you use an interpretation platform? So nowadays, more and more centers can afford to do NGS. And since its efficiency has been proven in choosing the right treatment for your patient, there is no real reason to dive in. However, the interpretation of the results can be quite complex. The different technologies, panels, testings, it can make it difficult to have one standard solution, especially since once the variants have been called, they still need to be interpreted, and that requires a lot of research to find their correct impacts with regards to the cancer type, the related drugs, their impact, and keep all this information up to date. And after all of this, you will still need to generate a report. So all of this is needed if you want to have all chances on your side to provide the right treatment, but it can be very time consuming. So for this reason, we believe that there is a need for a single tool allowing you to analyze and interpret the data, then generate a report and give you the possibility to still modify and share it. This tool should help all the people involved in the process. So the pathologists to continue to set them at the center of the diagnostic, the bioinformaticians to automatize their routine work for the reporting and use their skills to develop new algorithms for more challenging problems. The molecular biologists to let them having more time to develop and validate new panels, new tests, and then obviously to offer the oncologist an up-to-date and integrated solution to facilitate their decision based on a robust system of reporting. So at OncoDNA, we have 3 offers depending on which parts of the process you would like us to take care of. So as I explained, we have the kits. So if you want to send us the samples and that we take care of the sequencing, we can also work with the raw data that comes out of your sequencers. So we can work with BAM files, FASTQ files, etc. We'll come back to it later on. And we can also start from the VCF files if you want to still do the bio-IT analysis yourself. During this presentation, I will obviously focus on parts 2 and 3, since we are talking here about OncoKDM. So more in details, here's the process, the complete process. So you would provide us with the raw data, so the BAM files or the FASTQ files. We do a quality control check, and then we start processing the data. So we align it, we call the variants, add our annotations, and we can there calculate the MSI, TMB, DLOH, the CNVs. We also do what we call the alpha list, but I will come back to it later on. Then we clean the bias and send all the results to our specialist, who will interpret the data and provide you with the final report. So this report will be published on the platform that you can access, share with your colleagues, and there you will be able to do all the modifications that you want before validating the report and providing it to the oncologist. So I will give you a short demo. It's easier than explaining the different features. If you have any question, again, don't hesitate to send them over through the GoToWebinar interface, and we will answer them after the presentation. So here it is. This is obviously the final goal of OncoKDM, is to have this comprehensive summary that spans across all the information that you could have in the report. But let me show you how it works. So basically, the comprehensive summary goes through all the data that we had. And on the bottom of the page, you have the list of treatments that are split among those 4 categories, that are the ones with clinical benefits, the one with a lack of clinical benefits, the treatments that are associated with toxicity, and then the ones for which you might need to have confirmation. The effect is still undetermined. But let me show you the whole process. So first of all, OncoKDM is web-based, so you don't need to install anything on your laptop. You can share it directly with the link if your colleagues have an account, obviously. And just as a reminder, we work on somatic cancer for solid tumors at the moment. So in the first page, you have here a list of all your patient cases. For this one, I will go through this colorectal adenocarcinoma. So this is the page grouping all the samples and follow-up of your patients. So you can really have a view on the timeline and on the different follow-ups and the different analyses that you made with this patient to really have a view on its history. So here it's really the landing page of the analysis. You can see all these widgets that present the information that will be available in the final report. So one of the main goal of OncoKDM is really to integrate all of the information that you can have into one place. To check for which patient case you had this specific mutation, to do some statistical analysis, really just to have a look, a closer look at the bigger picture of all your patients actually. And so it is presented like this. We have basically one row per. Move it here. You have basically one row per mutation and for each patient. So in this case, we had all of these mutations, and you can see here for which patient and in which case. So that's it for the platform. I will now leave the floor to Jordi, who will tell you about the AGATA project that the Solti Group did with us.
Well, hi everybody. My name is Jordi Canes and I am Scientific Manager at Solti. I want to thank OncoDNA for inviting me to be part of this webinar and give me the opportunity to share with all the attendants the Solti experience with OncoKDM solution. Here in the screen, you can see this code where I state that OncoKDM allows us to include all the data generated during our AGATA study, integrating different technologies and bringing us a wider vision of tumor biology. To better understand how we get to that, during the following 10 minutes, I will introduce you all to Solti, to the AGATA study, and explain you our collaboration with OncoDNA and their solution, the OncoKDM. I think I don't want to see this. Okay. So first of all, just a few words about Solti. Solti is a non-profit organization dedicated to clinical research in breast cancer. Established in 1995, the clinical trials performed by Solti are designed to answer questions of major scientific interest and clinical relevance about the different tumor subtypes of breast cancer and its response to new treatments, as well as to convey proposals for new molecules. We have a network of health centers and more than 300 professionals involved in clinical research in breast cancer. More than 45 highly qualified professionals working in our office to manage the clinical trials that we perform. And nowadays, our science network is represented in 4 European countries, mainly in Spain. Now I want to give a brief summary about the AGATA study to put everybody in context and to understand the reason for the collaboration between Solti and OncoDNA with the OncoKDM solution. The mission of AGATA project was to implement a molecular screening program in Spain for patients with advanced breast cancer to improve access to clinical trials with targeted agents and to increase our knowledge on breast cancer biology. This is the workflow of AGATA study. You can see on the screen that archival or fresh tumor samples from primary or metastatic tumor were obtained from patients with advanced breast cancer after signed informed consent. After a quality control of the sample, target gene sequencing of well-known oncogenes and tumor suppressor genes implicated in breast cancer biology was performed using MySeq and EonTorrent molecular platforms analyzing different panels of genes. Results from sequencing were evaluated by an advisory tumor board comprised of medical oncologists, pathologists, and molecular biologists. Treatment recommendations were communicated to patient's physician, and in case of any actionable molecular alteration were found, the most appropriate clinical trial with any targeted therapy was recommended. After that, patients were followed up for survival until death. The current approach limits precision medicine to patients with actionable DNA alterations. And taking this into account, the AGATA team decided to perform in a second phase gene expression, protein, and methylome analysis to provide a therapy recommendation based on the full biology of the tumor. In AGATA, tumor samples were profiled in 5 different laboratories. Sequencing analyses were performed in 3 labs that used 3 different sequencing gene panels and platform covering 61, 35, and 24 genes frequently mutated in breast cancer. Only 7 genes were common in all 3 panels, thus, not all the genes were analyzed in all the samples. Gene expression assays and PAM50 calling were performed using nanostring technology. Considering all this, Solti and OncoDNA met and think about a solution for putting together all this data. One of the options that were considered at the beginning of the collaboration with OncoKDM, taking into account what it offered, was to use the platform to generate the report that would allow the molecular advisory board to make the decision according to the results obtained by the NGS analysis. As the study was already in advanced stage, including this functionality in a prospectively manner was not possible. Nevertheless, retrospectively, we compare the results obtained, validating them, and considering that it is an automated software, we obtain a faster search speed of clinical trials related to the alterations detected. Therefore, is an option to be included in a similar trial in the future to speed up the general turnaround time of the therapeutical decision. But in the end, how OncoKDM brought answers for AGATA program? Well, as in AGATA study, we generated data in different levels after genomic, gene expression, and proteomic analysis, we used OncoKDM solution to integrate all this data, and the OncoDNA team know how to perform a comprehensive report which includes these results and the interpretation for each case. Nowadays, in OncoKDM, we have included the 3 NGS technologies used in AGATA study, Illumina, EonTorrent, and Sequenom, integrating it with gene expression results by nanostring analysis in 172 cases. Our next step will be to add another level of data to these results with proteomics and see which relation can be found using these 3 technologies at the same time. Now I will do a demo to show you our solution that we work with OncoDNA. Well, you can see here that we have all our cases uploaded. I will search for the case that I know that we can show. And I think that here, to see the screen wider, I think will be better. Sorry. You can see here. It's okay. And then we have the clinical data of our patient that we loaded from our database in AGATA. The genomic results, the genomic sequencing results, and the alteration found in this case. It's the you can see that it's the same that my colleague showed you before. And we have all the functions that he mentioned. Then we also have the quality check of the readings. You can see it depends on the technology used. The drugs related to the alterations found. And here the impact and the status of the approval. Also some comments if anything. After that, we get the comprehensive summary. That is done by the scientific team of OncoDNA. Also, we have a bibliography related that is very useful. And if there is any clinical trial that is related to this alteration, but in this case we didn't have, so lucky. And also if there is in Spain, that you know that Solti is located in Spain, so if there is any clinical trial in here, but no. And after that, the additional tests in our cases is not related with immuno, but it's related with the gene expression. Also the analysis compared with the genomic results and the conclusions that we receive from OncoDNA team. And well, that's it, our demonstration. And to summarize, and to tell you after our experience with OncoKDM, we can say that We have obtained a platform that includes all our results, where they are treated in a safe and confidential way, with a great scientific team behind that collaborates with us to investigate the biology of breast cancer tumor and provides to us a tool where we can integrate all the data that we have generated from the tumor samples from AGATA study patients. And I want also to highlight that OncoKDM offers to Solti a web-based platform that can include all the technologies that we use, where we can obtain a comprehensive report and reliable data, thanks to also a technical team that adapts to Solti needs in an easygoing way. And for my part, that's all, and thank you for your attention.
Thank you, Jordi. Thank you, Nicolas, for this very interesting presentation of OncoKDM interpretation platform. Now is the time for question. So if you have question, please do not hesitate to use the question panel you'll find on the right side of your screen, and we'll try to answer it as best as we can.
Okay. So I see a couple of interesting questions from Sebastian here. So how is the database created? Do you use machine learning algorithms to extract information from publications? So no, we have algorithms going online every day to search for new publications and new information, but it's really our scientific team that will review these publications from start to end. So we know that we have some competitors who have algorithms searching like this, but we believe that there is a problem when working this way, is that you can have contradictory publications, for example. If you do some text mining algorithms, sometimes they will look only, for example, at the abstract, or you will look at the whole publication, but that can be contradictory. So we really believe that there is a need for a scientific expertise to decide on the actual classification of the variants.
Yes, we have another question from someone who is working and who is at work for now and can't, unfortunately, attend the complete webinar. Of course, we will share all the presentation, all the PowerPoint presentation, as well as the video recording of this webinar afterwards.
Okay, so another question, do you also provide information for clinical trials not listed in NCT? So at the moment, our algorithms retrieve information from clinicaltrials.gov, but we are working on new algorithms to retrieve information from different sources to try to gather really data about all clinical trials going on all over the world. So is OncoKDM included within OncoDeep? So OncoDeep is one of our kit solution, the solid biopsy kit. At the moment, if you use our kit, you will use the platform called OncoShare, for which the content is pretty similar to OncoKDM, but the interface is still a bit different. We are currently working on the integration. We will shortly move OncoShare to KDM, so you would actually have the same interface that I presented to you. How much do you charge per use, per sample, or per year? How much does it cost? So for this, it really depends on the solution that you would like to have. As I said, we can start from VCF files, but then you cannot have the very complete report. Meaning, for example, if we start from the VCF, we cannot have the quality control information. We cannot do the alpha list. Well, basically, it is more complete if we start from raw data. We can also develop some custom service depending on what kind of data you would like to integrate. But for all of these solutions, I would just advise to send an email to the email address on the screen, and we will come back to you with a proposition. Interesting one. What is the difference between OncoKDM and Sophia software? So, as I explained, I think the biggest difference is the way that we curate the database and that we annotate the variants. So Sophia typically use algorithms that will give some weight to the impact of the variants, but this is more of a black box, I would say, because we do provide all the sources for our interpretations, and we are really open to discussions. So if you use Sophia, you will have their interpretation, but you cannot really check if you do agree with it. And from my understanding, Sophia will give you more of a algorithm-based interpretation of the variants. So you will have a list of variants, the drugs linked to these variants, but we really try to have it patient-centric. You know, we look at everything, all the data that we could have for your patient, and we want to provide the best treatment. Also, I am not 100% sure that they do integrate. Well, I'm pretty sure they don't actually integrate other type of data than NGS. And we believe that it's really, in some cases, it's really important to be able to gather, you know, as I explained, NGS, but together with IHC, TMB, etc.
Okay, one more question for the CNV analysis. What are the minimal number of sample?
Where are you for CNV analysis? Okay, so for CNV analysis, as I explained, we just need to create the CNV baseline. So typically we need 10 samples that are negative for CNVs, and we will create this baseline. And then we validate the baseline with 5 samples that we know are positive for CNVs. And that's pretty much it. Afterwards, we can analyze the CNVs for each of the samples that were sequenced with the same technology and the same panel.
Okay, perfect. We have a last question from Jordi. Jordi, I will transfer the question regarding your experience with OncoKDM. Then you can read the question on your side. Okay, normally the question should appear on your screen, but I can read it. Now, with the experience of OncoKDM, would you like to proceed with the usage? Would you recommend it to others? And what are the 3 most important reasons for that?
Well, to us as an academic group, and you know that the resources are limited. Here, when we perform this trial and we get all this data, working with OncoDNA, with this solution with OncoKDM, give us a solution, and it was very helpful to have your team to work together with us and to have all this data integrated in your platform, because thanks to our collaboration we get this. The most important for us, I think that it was the point to have the opportunity to have all this data together, and to have the opportunity to, you know, to analyze it with you, and the facility that this platform gives to us. So for us and for our experience, for those groups or people that need to have all this data together, it's absolutely recommended to use it.
Thank you, Jordi. This will close our webinar session. Don't worry if you didn't answer to your question. We'll do it as soon as the webinar will be closed directly by email. One last thing before closing the webinar, I want to share you the link where you can download our brochure. And I would like, and we'll be very happy to welcome you in our OncoKDM community group you will find on LinkedIn. I will share the link to download the brochure in the chat panel. Then you can be able to download the brochure. One moment. Okay, I put the link on the chat panel, and here's the link. Okay, that's nice. And this will close our webinar session. Once again, thank you for the massive attending, and we hope you enjoy this webinar. And we are already looking forward to welcome you for our next webinar. Bye bye.
Thanks.
Thank you all. Bye.
Bye.
Thank you.